Good morning, I'm Vincenzo Randazzo and as a PhD student I'm part of the Neuronica Lab of the Department of Electronics and Telecommunication under the supervision of Professor Pasero and Professor Cirincione. I do my research on offline and online neural network applied to pattern recognition of big data and Internet of Things sensor data. More generally, in these years I've studied and deepened the field of data analysis, neural networks and pattern recognition and their application to biomedical data and medical devices for telemedicine and e-health. Among the several topics I've studied in this year, today I've chosen to present you the most promising neural network developed entirely by me and the research group I belong to, the GHX neural network for hierarchical clustering and clustering. I will also show its application to gene expression analysis of cancer DNA microarrays. In this graph, you can see the response of cancer cell to drug treatment. Each column is a tumor. If it's positive, the tumor grows and it is insensitive to the drug. If it's negative, the tumor regresses, responding to the drug. On the left, in red, about one-third are clearly resistant cases. On the right, in green, about 20% are responsive tumor, which regress in the presence of the drug. So, we asked ourselves, why tumors colored in red are drug resistant? Can we identify them before administering the drug? Since identical tumors do not exist, can we identify predictable similarities, that is, clusters in data, that can be exploited to instruct diagnosis and therapies? At this purpose, we worked on MCRC PDX transcriptional dataset made by Cancer Institute of Candiolo, which consists of DNA microarray with expression of more than 20,000 genes in around 400 million tissues. Each tissue has associated discrete variable describing its response to drug, as three classes: regressive, stable, and worsening cancer. The proposed dataset is quite complex to be analyzed because of its high dimensionality and due to the fact that genes correlate and influence each other. As you can see in the small portion of the gene expression dataset shown in the figure, rows and columns cannot be clustered in a simple way. For this reason, we have chosen to use biclustering to tackle this problem. A bicluster is a subset of genes and a subset of sample with the property that the selected genes are co-expressed only in the selected sample. By selecting samples and gene, a bicluster represents condition-specific pattern expression. Two-way clustering search for biclusters with constant value on rows or columns and with coherent values respectively. It can be proved that the numerical rank of the corresponding submatrices can be used as a figure of merit of the quality of the bicluster. The HCC index shown in the slide has been chosen for controlling the quality of the bicluster, as it also takes into account the noise in data. This index decreases as the value in the bicluster tend to be constant, differing for a constant on the rows or a constant on the column. To analyze the dataset, we create the new neural network for hierarchical clustering. GHXin builds a hierarchical tree based on another neural network we created before called SGXin. It is an incremental, self-organizing, top-down DCIF technique in which all data starts in a single root node and then splits are done recursively until all clusters satisfy certain conditions. Its novelties are the three techniques that follows: a double vertical grow to detect connected components within the network graph, an isotropic threshold mechanism to better approximate data manifold and neural neighborhood, and an outlier data relocation mechanism to reassign data that were wrongly labeled as outlier. GHXin has been applied to two-way cluster the gene expression dataset. Considering the very large number of genes, the fast hierarchical clustering is in this space. The figure you see on the left shows the parallel coordinate plot by visualizing genes and sample and murine tissues as featured on a leaf of GHX in the gene space. Blue polylines represent all genes available in the dataset, while red polylines stand for genes collected in the gene cluster. The red grouping of polylines show coherency, thus confirming the quality of gene clustering. A similar validation analysis is used after the second clustering in the tissue space, as you can see on the right. Even in this case, red groupings show coherency, confirming the validity of the approach. As a biological feedback, the scientific relevance of the selected genes has been taken into account. Among all the biclusters found, the one that grouped the most interesting genes in the cancer field has also the lowest HCC index value. It groups 7 genes of two families: the CSEC family, that is well known in medical literature as is associated with chondrosarcomas, but these genes are also found in normal tissues, and the Magia family that represents the melanoma antigens. These gene families are not all important by themselves, but may also co-regulate each other. It is also important to notice that this bicluster phenomenon has been observed within the tissues belonging to the third class, to which tissues unresponsive to drugs belong. In conclusion, we can say that GHXin has proved to be a valid tool to find interesting and unknown expression patterns in the gene expression dataset without requiring any human instruction. And the three goals we pose ourselves may have found a way to be tackled.